Good afternoon, everyone. Thanks for joining us today for this seminar. Really honoured to have Professor Andrew Ewing and Dr. David Joffe speaking today. Andrew is Professor of Chemistry and Molecular Biology at the University of Gothenburg in Sweden. He's a Knut and Alice Wallenberg Scholar, an elected member of the Royal Swedish Academy of Sciences, the Gothenburg Academy of Arts and Sciences, member of Swedish Science Forum, member of the John Snow Project editorial team, and chair of the Independent Long COVID Working Group. He has pioneered small volume chemical measurements to quantify the contents of individual neurotransmitter vesicles and their interaction with proteins. So, Andrew, if you'd like to come up, and I think David will speak after that, and then Andrew will speak again at the end. Thanks.
Thank you very much, Raina. It's a pleasure to be here and to give this talk. It's a wonderful, beautiful institute. And I think what I want to do today is, that was a wonderful introduction because it says exactly what I'm going to talk about from first and third parts of this talk. We're going to try to go through this. We're going to tag team. So I'm going to start out and talk a little bit about this Delphi study that we did that we'd like to spread the word about. It's 2 years old now, but it's very contemporary, as I'll show you. And then David will talk a little bit about dopamine and senescence and sleep and some other issues that are really cool in this topic. And then I'll come back to and bring us back down to smaller subjects with vesicles and so forth. So I'll start out with the Delphi, but let's talk about long COVID. This is a plot from a few years ago, okay, really from 2024, but 2023. And on this plot, so in 2023, they estimated that there were 409 million cases of long COVID. And as I'll show you from our definition issues, that this is probably a gross underestimate. And in 2024, 6.4% of US adults had long COVID. And so there's 20% of those with symptoms affecting daily activities. If you have 450 million worldwide, that's 4% of the world. Reinfections increase the prevalence considerably. This is almost certainly a low estimate, though, and it's really important to realize that. And here's a big issue, okay? Long COVID, the definition that Al-Ali in their paper, which showed that graph, was that long COVID represents the constellation of things based on symptoms, okay? And so what we found in the consensus was that it's the functional impairment, reduced effort tolerance, and so forth were important, but this is a definition that has to have a distinct clinical category regardless of the presence or absence of associated symptoms. And there's a lot of underlying things that happen with people after they have COVID that are just not showing up symptomatically. And I think this is really, really important. We did this survey of, I think, 23 or 24 countries were represented. We had 179 doctors and researchers, physicians and researchers that were polled over 3 rounds. By the end, it was 179. It started out on a smaller number. And you can see that there were a wide range of different professions that were surveyed. When we did this, we looked at definition, diagnosis and clinical assessment, treatment, evaluation of treatment, general research, organ damage, which is a big topic, of course, that we've published on as well, research on children and young people, vaccines, to a lesser level, but was important, and funding, economic and societal issues. And you can see the numbers of statements and whether there were consensus. And the consensus was pretty good. I mean, consensus, there's only one statement that had unanimous consensus, but that statement that had unanimous consensus was that physicians need to be better educated. That was a really important statement, okay? But there were a lot of others. And so when we summarize this, okay, this is a global reach, okay, urgent consensus, okay, and we really call this was a wake-up call for governments and health systems. Unfortunately, they have stayed asleep, okay, But the 10 urgent areas of consensus on this, okay, are that educating frontline providers is non-negotiable. That was unanimous across everyone who was polled. A universal definition is essential. Long COVID is not just tiredness, that there's all these other things that take place, including serious complications in the immune system, the brain, and different organs, okay? Even when conventional tests appear normal. And that leads to long COVID is often an invisible disability. Not always, okay, but often. Many long COVID sufferers experience disabling symptoms that don't show up on routine tests, and this makes it really hard for physicians and healthcare workers trying to diagnose this. Current diagnostics fall short, therefore, a syndrome of syndromes. There's clear consensus that long COVID is not one disease, but a cluster of overlapping conditions. And this is sort of where this deconvolution with ME/CFS and MCAS and so forth comes in. And there's a lot of people that get really upset about this, but the fact is, this is the way it works out. The pathology is different, but there are associations, and we need to recognize this. Children are also at risk, okay? And this is the biggest myth of the pandemic in my mind, okay, is, other than that it wasn't airborne, is that COVID was completely innocuous to children, and this is just a lie. And so we have clean indoor air, because there's a lot of people. It's really interesting because a lot of pediatricians who were so adamant that it was not affecting children early in the pandemic have now come over to the other side and are working on long COVID in children. And this is really a testimony to their ability to say, wow, we were wrong. Let's go for the right thing here. And I'm proud of a lot of these people. Clean indoor air is a prevention strategy. It's not just masks, guys, online, okay? It's clean air. Right now, we're sitting in a room that's 400 CO2. That's ambient. That's what's outside. And I'm assuming it's probably filtered on the way in and out. And so this is a very safe environment. We really don't need masks in here, okay? Okay, and if you want to shoot me for saying that, go ahead, shoot, okay? But the real reality of it is that's what's safe. We need global investment now. That's pretty obvious. And the economic fallout is massive. It talks about it in the paper, but there's enormous amount of money that's going to be spent on this over the next decades. Okay, so at this point, I'm going to let David talk and introduce us to dopamine, senescence, and sleep before I come back and talk a little bit about more dopamine in vesicles and cells.
Okay, so just before I introduce David, also I'd like to acknowledge the traditional owners of the land that we are meeting on, which is the Vesicular people of the Eora Nation, and pay my respects to elders past and present, and any Aboriginal or Torres Strait Islander people here today. So David Joffe is a sleep respiratory physician who holds a VMO appointment at Royal North Shore Hospital. He's the vice chair of the World Health Network Long COVID Experts Group, with whom he's co-authored overarching papers on the multi-system involvement of COVID. He has extensive clinical experience in the evaluation and treatment of long COVID and collaborates widely with his extensive international group of long COVID experts. He's co-authored several papers on long COVID, including the award-winning article, Vascular Pathogenesis in Acute and Long COVID, Current Insights and Therapeutic Outlooks, published in Seminars in Thrombosis and Hemostasis. David, welcome.
As always, it's a wonderful pleasure to return to my alma mater at University of New South Wales. It was a great pleasure to actually bring Andy to my spiritual home, even though I'm obviously a Sydney University person. So what I wanted to cover were a few things, a bit of clinical and then a little bit of, you will find out as we are transitioning that despite our wonderful passion and friendship with the WHN, as a group, we just kept growing and growing, and we started to have more and more international people. And so with the launch, or with the meeting that we had planned in Amsterdam at the end of this month, we are going to be launching the International Society for Long COVID. And of course, post-acute infections. The focus really, Andy will talk to it, is to bring as many people as we can together to really focus on this global catastrophe. So this was really a seminal paper from 2023. It's now 3 years old, and was really what I wanted to take to Andy, because he understood this better than I did, and I wanted his opinion when I went to Sweden. This was an amazingly well done study with human stem cells that they actually grew, HPSC dopaminergic cells that are permissive to COVID-19. And so what they were trying to do was work out what was, permissive to infection and that wasn't. But the fact is that whilst people thought that the frontal lobe didn't have ACE inhibitors, so it wasn't a problem, well, yeah, the virus didn't worry about that. It used Empress instead, and so it found its way in. By the time you've been infected, within 2 hours, the virus has gone through your nose and into your prefrontal cortex. You haven't even sneezed yet, it is already in your brain. That is work from our friend Danny Beckman in macaques. So what they did was try and identify, using immunofluorescence, specific dopaminergic cells. That's what these guys were trying to do, and then try and work out how they got in. It's a little bit complicated, but I'll try and go through it. So they purified these cells and then isolated them and infected them with COVID. And not surprisingly, they were then able to do a PCR and demonstrate that the virus wasn't just there, it was actually replicating. So people who think that, replication and persistence are not, on the cards, we've found it everywhere we've looked. And wherever we've found persistence, it's there and is almost certainly, in my own opinion, replicating. But it may not be replicating in a hurry. They looked for the nuclear capsid protein, so they knew it was actually the virus, not just spike. And they confirmed that at least 8% of the cells at 72 hours were basically infected. They tried to block it with an ACE inhibitor to block infection, and that was perhaps not terribly a good idea because there wasn't really enough infected cells by then. So they had a mock infected arm, which they used as the control, and infected dopaminergic cells that showed a distinct transcriptomal space that was compared to the controls. In other words, these cells are very different to the others. What they also found was that there was a kind of subtype of specific vulnerability in these highly specialized dopaminergic cells. And what they found was there were certain clusters within the cells that had dopaminergic markers for specific entities, meaning that it doesn't target absolutely all and every cell, but it is targeting ones that it can get at. But what's important is that A9-DA is a crucial marker. And that one in particular is really important. And the reason I say that is it is highly represented in the substantia nigra. And that is obviously, if you like, the home of Parkinson's, but I will explain a little bit of why I think it's more. This is really just to demonstrate the difference between the mock and the real, and to show you that, there was mock and there was very real injury. And so what they actually showed, to put it simply, is that what the virus does is not specifically and directly neurotoxic, but one of the things it really does is that within these cells is a thing called senescence-associated secretory pathways. Pretty much most cells, but certainly these cells, have a recovery mechanism. So in the circumstance where cells are attacked by your own immunity or by a virus or bacteria or whatever, there is some way of the cell to recover from the insult. And one of the problems is that the virus specifically targets the recovery pathway. So having perhaps damaged the cell or having perhaps just inflamed the cell, the way in which it actually makes a mess of it is by preventing it from recovering, hence the term senescence. In other words, these cells may not die immediately, but they certainly are going to die faster and earlier. And so when they looked at them, what they saw made a lot of sense. These are bigger cells. They can't recover. They can't get back to their normal morphological conformity. To give you some idea about what it looks like, so when people say there are no tests, I'd argue that we just can't afford to do the ones that matter. This is 50 patients from a long COVID neurology, specifically neurological clinic. In other words, those with fatigue and brain fog. They match them, so it's pretty early data. This is all, you know, much of this would be, you know, unvaccinated, some vaccinated. What they compared them to was interesting, was MS patients who also have fatigue, but in their circumstance, it's a very highly specific autoimmune disease that drives demyelination. And they looked at MRI, and they divided them up, and they looked at the severity of the fatigue. What they found was that the quality of sleep, which is the Pittsburgh Sleep Quality Index, were really substantial. What I wanted to point out is that this, which is another sleep metric, the Epworth Sleepiness Score, named in honour of the Epworth Hospital in Melbourne by my dear friend, the late Murray Johns, is an extremely valuable, validated tool, not for sleep apnea. For that, it's pretty hopeless, which has always been my opinion, and as was certainly Murray's, but it's a wonderfully well-validated tool for narcolepsy. And so when you are seeing those with a metric of 15 or more, you shouldn't be thinking about apnea or anything else. Your first thought should be narcolepsy, which is why we still include it in our sleep lab. So when you see someone has a normal sleep study, but they've got an Epworth of 19, you shouldn't walk past that. You should go back and have another look and think about looking for narcolepsy. What was also specifically interesting was the neurocognitive assessments. The damage to these people with COVID fatigue, for want of a word, brain fog, was both auditory and visual. But there wasn't really an issue with accuracy. So it's more a kind of visuospatial, which we know is a kind of frontotemporal domain. One of the big issues with patients I see is that I want to drive. They have significant word finding, word replacement, very frontotemporal dementia kind of characteristics, but in particular geospatial. They get lost, they lose things, they change words, they forget words. As you can see, there's a substantial shift in sleepiness and in sleep problems, and of course, not surprisingly, depression and anxiety. But when you look at the actual brains, and I guess this is really what I want to show people, is that, you know, short-term memory, they show lower thalamic volumes, inward deformation of the left thalamic cortex, altered anastropy of the left thalamus, aberrant thalamic diffusion with physical fatigue. So they could map using really, fabulous software, areas of the brain that are damaged. But what I really wanted to point out was it was the basal ganglia structure. The volumetric analysis pointed to the base of the brain, the putamen. The clinical psychologists and psychiatrists have often pointed to the putamen as potentially the home or one of the homes of ADHD. I'm seeing an awful lot of ADHD in adults who I don't think have ever had that. It's a new diagnosis for them. They come along and they've gone and seen someone and they're sleepy and they're tired, and so and I can't concentrate and I forget things and I have inattention. So they say, got ADHD, and put them on a stimulant. Well, for some, they've actually got narcolepsy, so it might be the right direction, but for many, it's the wrong label. And I just want to point that out, that, there's a sudden reason why we are allowing GPs and nurse practitioners to prescribe amphetamines, because we don't have enough skilled psychiatrists to make the diagnosis. Why now? Well, we're 7 years into a brain-damaging virus where we're now seeing this. And in kids, I thought it was just me when I spoke to my UK colleagues. Ray Duncan in particular said, oh my gosh, I'm seeing the same thing amongst cardiology patients and long COVID patients. Most importantly, though, was the sleepiness. There's a big difference between fatigue and sleepiness. I kind of spend my whole day doing that. It used to be easy. It's no longer. I get people sent to me for queried sleep apnea, but when you take a history, they not sleepy at all. They just have PEMS, post-exertional malaise. It's not that they sleep, they just can't get out of bed. And so when they go to the GP and say, I can't get out of bed, they go, oh, well, you need a sleep study, you must have sleep apnea. Well, it's not what they have. Although we do know the incidence of sleep apnea at 4.5 years is twice that in those who had a mild infection. The driver is weight gain. Just saying. I'll just skip over that. The white matter, interestingly enough, is not affected. So it's a distinct structural change. None of these patients had been hospitalized. They were all young, not what you want to see. Severity was independent. That's one of the problems. You get a mild infection, doesn't mean you get a mild injury. It's not the same. And that the COVID fatigue was really associated with depression and, here we go, sleepiness. Sleepiness is the hallmark of narcolepsy, which is a dopaminergic injury. The drug of choice we use is a drug called modafinil or armodafinil, which is a specific dopaminergic agent for wakefulness promotion. And so what they were getting at, just to point it out here, is dopaminergic imbalance. And so this is what Andy and I were at length talking about and how we really became friends, because myself as a clinician with an interest in Parkinson's and a genetic predisposition to neurology, I've had a long fascination with Parkinson's and in particular REM behavior disorder. And so my dad, who's now retired, but he was the guru of Parkinson's, and I had an ever present stream of guys who had Parkinson's who would jump out of bed and do strange things. Likewise, I had a lot of veterans, both from the Gulf War, other wars, every other miserable place we send good people. And I would see these guys and they'd be sitting there and they'd be doing this. And I'd say, what's that? And they'd say, oh, I was in the clinic. They said it was my nerves, doctor. And so I'd walk across the hallway and I'd get my old man and he'd come in and go, yeah, that's a pyramidal. I'll see him next week, extra pyramidal. I'll see him next week. And of course, you know, there's a 40-year-old guy with Parkinson's. It's not your friend. The younger you are, the worse the prognosis, just to say that loudly. So this is the thought that I've had. This is a culmination of decades of seeing veterans who, in all honesty, have probably taught me more than I've taught them. At the bottom of your brain, below your pons and above your spinal cord is a big long tract, which is the reticular activating system. It is the center of your consciousness. If anybody ever asks you, how do you know you're a sentient being? The answer to that is, my reticular activating system told me so. It is that, if you like, which we remove chemically when we give you a general anesthetic. So we can chop you into pieces. It is beyond or below stage 3 or 4 sleep. You are actually comatose. That is, if you like, where coma exists. On one side is the center that controls REM. There are 3 absolutes. You are either awake or you're in REM. There is no in between. It is entirely serotonergic. It is one of the fascinations of REM. And probably the reason for that is it is when we lay down memory. So while slow wave sleep is crucial for washing out your brain, in other words, glymphatic function, we also lower our heart rate, lower our blood pressure, and we actually wash out our bodily wastes, our lymphatic system. And so slow wave sleep, if you like, which is the crucial component of sleep, is really about getting rid of the waste products. REM, on the other hand, is very much about attention, concentration, and specifically short-term memory. It's when you put down memory. One last fact is that you are completely paralyzed in all your voluntary muscles. Any muscle over which you can exert a physical control is paralyzed. And so you will still breathe, your bowels will still move, your heart will still beat. But anything that you can move yourself has no electrical signal. And so one of the tests we do in our lab in long COVID people is to put 2 extra electrodes, one on the forearm, the other on the thumb. Those are entirely voluntary. They will not move any other way. To look for whether the electrical signal in those muscles disappears in REM. it's called a SINBAR montage. And so every single patient that I have with long COVID gets a SINBAR montage. What I was interested in is, do they have normal paralysis of REM, or do they have REM sleep without paralysis, which is, if you like, the precursor to REM behavior disorder, actual physical functional movement. On the other side is the locus coeruleus, which for the Greek scholars is the blue body. The reason it is blue is it stains heavily for copper, which is all noradrenaline. This is your flight and fight and fright center. It has a projection to the substantia nigra. For years, we would look at brain scans and say, well, the substantia nigra has disappeared, it's Parkinson's disease. And that was true, except that the Israelis actually demonstrated years thereafter that the injury starts down here. And it's really almost as though the abnormality is here, and that these cells literally just die as a consequence of an abnormality in this pathway. And so that's the crucial thing, is that this system is related to Parkinson's and also to dementia, the Lewy body. And so it's really important. Not only did they identify the pathway, they even made a medication for it. But coming off this as well is a fascinating pathway to the amygdala. The amygdala, if you ask the psychiatrists and psychologists, will tell you is the center of anxiety. I actually am not a big fan of the word anxiety. I think that has connotations. I prefer the word hypervigilance, and that's the term I use for my veterans. These guys are not anxious. I mean, my goodness, they've got DMSOs and VCs and absolute, nuts of steel. I mean, these guys are soldiers. But they are hypervigilant. They do not go window shopping. They're always looking in the window to see who's behind them. When they drive, they know the color of every car in the rearview mirror and on which side of them. And you certainly do not want to tap one of my SAS guys on the shoulder without giving him a warning, or you might find yourself with a very, very smacked up nose. They are hypervigilant, not anxious. But the reason that we put a memory down there is very important. It's a negative encoding memory. What is crucial to recognize is it is dopaminergic. It is, in fact, the amygdala that is the center of compulsivity. It is what drives everything from addiction to compulsivity and other sorts of behavior. And it's no surprise that within a week of the virus being in your prefrontal cortex, Danny has demonstrated, yep, there's cell-to-cell spread straight to your amygdala. It barely gets clobbered. Red berries in the cave. The reason I use this analogy is, why would God in his or her infinite wisdom design something like this? Well, I'm glad you asked, because it was a question I often asked myself, and then it dawned on me. We still live in caves. So essentially, REM sleep is a mammalian land adaptation. If you look at monotremes, and so a wonderful, brilliant American came here to work with echidnas. We are the only place where we have mammals that are egg-laying. They are somewhere between a reptile and a mammal. And he demonstrated with brilliant science, not that I would want to go anywhere near an echidna with needles, but he did, and actually put electrodes into them and demonstrated a very primitive form of REM sleep in echidnas. And that was years ago and always stayed with me, that this is a really very primitive network for us and others. So why? Well, I guess to put it bluntly, if you are sleeping in a cave and a bear does roar, well, you want to go from being in REM to being awake. You definitely do not want to be paralyzed, and you most certainly want to be a sentient being. You want to fire this thing off so that you will flood your muscles with blood, you will shut off your sphincters, and you will run the hell out of there. I guess this is called natural selection. It's fascinating. Patients with Parkinson's often have trouble with initiating movement, particularly when they get to lines and other things. And, I must have asked neurologists 1,000 times about that, but it's this, in essence, is that there's a trigger abnormality that means that they're going to be barefoot. But why does it send it here? Oh, well, that is so that when you run from that cave, you're going to keep running. And you're going to keep running and running. And I can promise you one thing, you are not going back to that cave. That's the bear cave. That's the negative enforcing message. We know if you give red berries to primates and make them sick, they would rather starve to death than eat red berries again. We're the same. We just like to think we are spectacular in some way or form. But God only made one motherboard and pretty much one unit of RAM. Just put in a bigger unit of RAM and a bigger hard disk for the rest of us. But fundamentally, this is the motherboard. And there is only one, and we share it with most species. People don't realize that even jellyfish sleep in a form. They have a period of quiet, no movement. Everything has to sleep. And so when you are asleep, you're vulnerable. And it is in this period of vulnerability that you need to maintain some level of vigilance so bears don't eat you. And so it's crucial to understanding how these pathways are crucial when we're talking about a virus with dopaminergic injury. Because, yes, I am seeing all of these things. The incidence of restless legs in both acute COVID and in post-COVID is around 40%. That's a dopaminergic injury. Parkinson's. You see people with Parkinson's who've had COVID or those with early cognitive impairment. The evidence is overwhelming that what may have taken them 6 years to reach a point of needing care will take them 6 months. I've seen that in many of my own patients, which is really very tragic. People who, were, eventually going to, meet their, milestones for dementia. They get COVID and they can't remember things and they are in trouble. But PTSD, and so that was my fascination. These are my veterans. This is their life. But these things all overlap. I have guys with PTSD who now have narcolepsy. I have Parkinson's patients who have profound sleepiness in a pattern that is almost identical to narcolepsy. Obviously, restless legs is a feature of PTSD. 80% of veterans have PTSD. A substantial number of patients with PTSD, almost tenfold, are likely to get Parkinson's at a young age. Major depression. Well, major depression also lives there. And of last but not least is REM behavior disorder. And so REM behavior disorder, where used to be a condition first described by men, or in men over the age of 50, Mark Mahowald studied in Minneapolis many years ago in the 80s. What he demonstrated was if you watch them long enough, 80% of them who had clear dream behavior disorder or dream reenactment behavior disorder, 80% developed Parkinson's at 80 years, at 14 years. The problem for me is that I have young people, long COVID people as young as 16 up to 46, and they are women as well as men. They are not men over the age of 50 who have demonstrable REM behavior disorder. We haven't got to 14 years for me to tell you what happens. I just put it out there that it's something we should look for. It's something that's of concern. It's a marker of dopa injury, and there's reasons to understand that. I'm going to let Andy do his thing. Thank you for your attention.
Okay, thank you. I'm going to take us to a different world now, the world of chemistry and going down into cells and subcellular regions, and try to lead to a hypothesis again that I have about what is happening at the molecular level with COVID and cells and changing dopamine. And I'd start out by saying that David mentioned modafinil in the middle of his talk, and we've studied modafinil about 5-6 years ago. We published a paper showing that it affects neurotransmitter vesicles, okay? And this process of exocytosis that's shown in the upper left corner, it affects this process, it slows down the release process, and it changes the amount of dopamine in vesicles. And so this is a really interesting lead-in or segue into what I'm going to show you in the next few minutes. We've developed techniques using electrochemistry at small carbon fiber electrodes to measure this exocytosis process at model cells and in vivo as well. And we've also developed techniques to measure the contents of vesicles over here on the right side, with the vesicles coming up and absorbing to and opening on the electrode surface. So we can measure the amount of material, the amount of neurotransmitter, in this case, dopamine, coming out from the vesicles during exocytosis and in the vesicles, using electrochemistry, looking at this example of the response and measuring the charge to give you the number of molecules. So we can actually look at these vesicles, and we discovered in the process that the Katz's principle for all vesicles is incorrect. And the principle is that all vesicles open all and dump out all of their contents during exocytosis. This is basically incorrect. And we discovered this. It was really discovered, of course, a series of papers before this dating back to the late 90s, but no one really put it all together. Okay. One paper in 1997, I think they sort of got it, but nobody believes it. And still a lot of neuroscientists don't want to believe it. Looking at a model system, which is the first place we went, PC12 cells, a lot of neuroscientists don't like this, but I'll show you more in a minute. We showed that about 63% of the dopamine was coming out during an exocytosis event. And so this is important because it speaks to the mechanism of this release process and to communication between cells, which comes back to this mechanism that's changing with modafinil, that's changing with COVID, as David just nicely pointed out. And so we went on and spent the last about 8 years showing that this was a pretty ubiquitous process, that it's across all the cell types that we've looked at so far. There's one case in the fruit fly, Drosophila, where we found that a subset of neurons release 100% during exocytosis, but all of the cases are a percentage. So this is really a different way of looking at neuroscience. And it's important because this process of partial release, which is the predominant mode of exocytosis, there's also something called kiss and run. There's also full release. These do happen, and we've studied these, and I don't have time to go into it all in detail, but about 95 to 98% of what's happening during exocytosis is this partial release. The vesicle opens, dumps out some content, closes again. It's kind of like pulling a trigger on a squirt gun, okay? It's very simple, all right? But it means that it can be redone, okay, that these vesicles can be reused. It also means that it can be modulated. And so if you look at a neuron and look at a synapse, the synapse has release to communicate with the next neuron, okay, here. And this is all the things we're talking about in places like the caudate putamen, okay, where the dopamine neuron synapses are, or one of the locations, as well as the frontal cortex and the nucleus accumbens, and so forth. And we've known for a number of years that one way to modulate the amount of dopamine transmitter in the synapse is by changing the number of events that take place. And this is basically done by changing things related to the snare complex. And the snare complex in 2013 was given the Nobel Prize in Physiology and Medicine because they discovered this as a way to modulate it. But it's not the only way to measure this, okay? Or to change this, to modulate this. The other way is to change the fraction that comes out. And if you change the fraction from like 5 to 10%, or even from 60 to 90%, okay, so one end of the spectrum or the other, you get a large change in that modulation of the process. This is called plasticity. This is how we change the vesicle, the strength of the communication between the neurons. And it's really important. Okay, so with all that as a backdrop and thinking about this in terms of changing neurotransmission, as David has just sort of amply shown us is important in COVID and long COVID, now let's look at the vesicular process. And one of the things that we discovered a few years ago, not with vesicles, but with stress granules, and stress granules are cells that are stressed in cancer cells. If you give certain types of treatments like arsenite to them, or if you give anti-cancer drugs to the cells, okay, like cisplatin, you create these structures that are made of, up in the right-hand corner as sort of a model of these. They're made of RNA, it's really mRNA protected by proteins around them, okay? And that's a major part of what is thought stress granules do, is to protect the mRNA during the time of stress. in many of these stressors, they don't last for very long. They're there for an hour or two, and then they go away. Although the chemistry in the cells, as my colleague New has shown very nicely, is changed for many hours afterwards. Okay, the chemistry in the cells. But there's a lot of things. But these stress granules, what do they do? Well, we accidentally, but really cleverly, by my postdoc Kiki He, who was in my group, discovered that H2O2, okay, hydrogen peroxide, is made by stress granules. Okay, and so this was a big discovery, and we were working with a stress granule expert, Nick Locker, who's on the bottom here, and this was pretty interesting. Each stress granule, just sitting in the solution, makes about 40,000, says 38,000 molecules. Okay, and we can quantify these with our electrochemical techniques. And so then we went on to look at how these might affect vesicles. So this stress to vesicles. And I'll bring this back because stress granules, they're a protein complex, a lot like spike protein complexes. Okay, so we'll come back to this. But if we look here on the left side of this slide, you see the frequency of events as we change. And these are the events, these events of stress of vesicles hitting an electrode. So we're looking at vesicles from cells. And we see that the frequency of the events drops down when you have stress granules present. So stress granules are changing the vesicles. And then if you look at the number of molecules in the vesicles, not in the stress granule, but in the vesicle, the number of molecules goes up with stress granules. And so we then went and looked, trying to figure out what was going on here. We went and looked at TEM of these cells, and we find many of the vesicles actually have multiple dense cores. Usually each vesicle has one dense core, okay? And I'll give you the numbers in a minute. And now we're finding a change. And in fact, if we looked at the relative number of events versus the size of these vesicles, there's a blip here down here on the right showing there's a subset of the vesicles that get larger. All right, and so what we did, and you can see that with the arrow, and so then what we did is we counted them, and it turns out when you have no treatment, that you get single core vesicles are 98% of the total, and about 2% of them have two cores. With stress granules present, it goes to 66% and it goes to 26% double cores and 8% more than two cores. Now, why would you get more cores? Well, it's the best reason you can come up with is that vesicles are coming up and fusing with each other and becoming larger. That fits with the largeness of the vesicles, the subset of vesicles having this, exactly with this. If you in fact take these percentages and simulate the shift in the relative frequency versus the log of the number of these molecules, it fits exactly with the simulation. I think I have a circle, yeah. And so we simulate this, it fits very nicely for the shift here, okay, in this sizes. So we're making multi-core vesicles with stress granules. And our model for this that we've proposed is that the stress granules connect with vesicles. They connect through some part of their protein structure on the outside, because homotypic fusion doesn't like to occur. If it did, we'd have a mess in our brain, okay? But these stress granules are causing these to come together, and their formation of the hydrogen peroxide is causing the membranes to become more able to fuse, okay? They're less stable, and you end up with multi-core vesicles after they fuse. And we've shown that hydrogen peroxide does this to membranes before in earlier work that we've carried out. And so a big question is, how do the stress granules generate the reactive oxygen species? And we found some papers, and I've been fascinated by Dick Zare's work at Stanford for a number of years. He's been since about 2018 or 19 when he first published, where he found that droplets in a vacuum formed H2O2 on their surface. And they're formed electrochemically, it turns out. And they've shown in recent years that if you take polymers and make conglomerates of them, a disordered polymer into a phase separation to get a conglomerate here, or aggregate, okay, what they call a condensate, a polymer condensate. What you find is that there is a potential field at the interface here. And this potential field, amazingly to me as an electrochemist, because we're talking about 0.6 of a volt or somewhere around there, that's how big it is. It's big enough to oxidize and make hydrogen peroxide by oxidizing water. Okay. Now, the question is this what stress granules are doing, and how ubiquitous is it? Okay. If it's ubiquitous among all protein clusters and aggregates in and out of cells, it's incredibly important. This is really going to be changing how things are functioning. But different protein clusters might create different amounts of ROS and interact differently as well. So that's all important. So we did a series of experiments that we published last year where we looked at the enzyme dominant pathway, which we originally thought it must be an enzyme in the stress granules causing the formation of hydrogen peroxide, versus the water dominant pathway. And if you look here, you find that these are electrochemical experiments that you probably don't follow exactly, but these are these spikes from these stress granules hitting the surface. Now we're back to stress granules, and there's no change with oxygen or nitrogen. Okay, but there is a change when you do things like D2O, which forms a different layer at the surface with the water, and DMSO. Okay, and so D2O, which slows it down because this is deuterium is heavier than hydrogen, reduces the amount of molecules, the H2O2 molecules you see, and DMSO, which loosens the hydrogen bonding, forms a larger amount of hydrogen peroxide from the stress granules. So this is really important, okay, because it tells us that we're making these at these protein polymer aggregates, okay, on the surface. Okay, so we come back to this idea of what happens to vesicles. Well, what happens if we do this, if the same thing is happening? And this is my proposed mechanism for this, and I don't have evidence for it yet, but we are working on this, okay, as we speak. And but what if the spike protein aggregates, and they do look like this? It almost looks like COVID, the SARS-2 virus, right? I mean, but these are aggregates. It could be the SARS-2 virus as well, but I mean, I think the aggregate's more likely. If they're making these same condensates, and it seems like they do make condensates, these could be attaching to vesicles and causing them to undergo homotypic fusion. So I think this is a really fascinating proposal as to what could be happening, which makes this consistent with what we see with modafinil and what we're seeing with the clinical aspects of dopamine systems with COVID and long COVID. Spike protein might regulate transmission via the ROS and the organelle morphology. Okay, and so this is a simple diagram of a cell, and it shows you that instead of having replacing stress granules, which we think do all of these things, spike protein might change plasticity, might change exosomes, might change mitochondria. It could have a lot of different things, and it's going to behave differently based on the amount of ROS that it produces. I think ROS is an enormously important part of what's happening at the cellular level with the SARS-2 protein, SARS-2 virus. Why would SARS-2 induced homotypic fusion be important? This is a long list. I don't have time to go through it all, but it would change the fundamental unit of neurotransmission, transmitter release, fraction released, etc. But if you go down here, it provides, number 10 is it provides a plausible mechanism for long COVID neurological symptoms and Parkinson's disease. If you go through the whole list is very similar to many of the things that David was just talking about in his talk. If you go, many long COVID symptoms are consistent with subtle synaptic dysfunction, not widespread neuronal death. And there was a lot of talk early on about SARS-2 causing cell synthia, okay? And this is a big issue. But I mean, in fact, really what's happening in my mind is that we're changing what we have rather than we're destroying it, okay? Or we're probably doing a little bit of both. But brain fog, impaired executive function, slowed processing speed, reduced attention, autonomic dysfunction, sleep abnormalities, and altered dopamine signaling all could be taking place. Perspective on this, if I was a betting person, I would bet that there's being a large increase in neurodegenerative disease in the future, and eventually it would be tied firmly back to COVID infections. And I think this is very much what Dr. Joffe was saying, okay? Honorary professor, right? You're an adjunct professor. Healthcare workers must be trained to recognize long COVID not just as post-viral fatigue, but as a complex multi-system condition that involves the brain, the immune system, and all the organs, the heart, etc. With that, I just want to acknowledge the people that are doing the work. This is actually a picnic, so we have families there. It's a little bit bigger than the actual groups. It's the two main bioanalytical imaging sensors and cells groups at GU. One is mine, one is New Fawn's group, and doing a lot of things related to all the things that we're talking about. The dog is not actually involved in the research, but is a pet, okay, just so that you know. And the last thing I want to do is I want to acknowledge Raina McIntyre again for hosting this seminar, but also for being part of the Delphi that I led this out with, okay? And not only was she one of the panelists, she's a co-author on the paper because she made major seminal contributions and kept us on the straight line, especially with vaccinations. So thank you very much, and thank you for your attention.
Thank you very much, Andrew and David. I might get both of you to come up. We've got a couple of questions. Was that the only one, the one you showed me online? 3? Okay. So, I'm not sure, maybe, don't know who wants to take this one. Do these changes occur to the same extent in vaccinated people who were not infected until fully vaccinated? So does vaccine give some protection against?
I think it'd be fair to say that early in the pandemic, when we had really well-matched vaccines, the Wuhan variant, certainly the ones through to about 22, 23. The XBB mixed one with Wuhan original was probably our biggest failure. The problem is that we were doing, I think, reasonably well, and certainly the evidence for vaccines offsetting long COVID was around 40% when Zihad Al-Ali published that paper, which was encouraging. But I think it'd be fair to say that at this point, with the evolution to JN1, which occurred at the end of 23, beginning of 24, that was a seminal shift. It was as close, it had 34 different mutations to Omicron. So it was as close to Omicron as Omicron was to Delta. And so at this point, the vaccines are valuable in terms of, I think, reducing severity of disease, certainly reducing death and hospitalization. But if I said to you that the evidence is that they prevent long COVID, I think we may be past that point, which is really important to recognize. Doesn't mean you don't get vaccinated. We should protect our elderly. We should protect ourselves from severe disease. But I think it's fair to say that the virus now is much more cunning, and it's not looking to blow us up and kill us. It's looking to eat us slowly, and that's long COVID.
Thanks, David. Are kids with long COVID being misdiagnosed as having ADHD or narcolepsy or other diseases, or is long COVID causing these conditions in kids? Maybe that's one for you as well.
I think it goes both ways. I think that there are kids who may have had COVID and now have narcolepsy. That doesn't mean they have long COVID. It means they have narcolepsy with a COVID-related associated neuro-autoimmune disease. ADHD, well, we've known about ADHD a lot longer than we have about COVID. The reason I pointed out is that there seems to be an epidemic of ADHD diagnoses occurring not just in kids, but also in adults. Now, I'm not a pediatrician, so I can't speak to them, but certainly in adults, I'm seeing an unusually large number of people who are presenting with this novel diagnosis, where really, when you take a good history, much of what they are complaining about are neurocognitive deficits that look more to me like a COVID brain injury than specifically to having just developed inattention. And I guess that's the dilemma, is that we can't separate it out entirely at this point. I think it goes both ways. I think there are kids being probably accurately diagnosed with narcolepsy, and clearly some well diagnosed with long COVID. And there's going to be a whole lot who have both.
Okay, we have another question here where I'm just going to summarize the quest, sort of focus on. So it's talking about exercise therapy, assuming simple deconditioning as a driver of long COVID, and how do we sort of switch to a more evidence-based treatment approach?
Well, I think we're fortunate to have had David Toller come and talk last time about some of the misdirection and some of the clearly erroneous nature of the studies undertaken using, cognitive behavioral therapy and graded exercise training and those sorts of tools. And, the people who preceded long COVID are those with ME/CFS who had tried to warn that, some will do okay, but many will not. Many will get crushed. And so the evidence certainly from Mark Fay's group, who's one of our group, and the muscle biopsy stuff from Rob Wust, who's also one of our group, is pretty obvious that there is something strikingly abnormal in terms of a massive inflammatory release that occurs after a very low impact dual-day exercise test. And when you look at Rob's data published in Nature, those muscles are not deconditioned. There are at least 8 different abnormalities, from microclotting to changes in fiber type, to evidence of nets, to inflammation, mitochondrial changes. There's a whole cluster of things that are not deconditioning. And he did the study to demonstrate quite clearly that there is deconditioned muscle in those who don't move. And then there's long COVID muscle. It's unique.
Thanks. We've only got literally a couple of minutes. Are there any questions in the room? Answered all the online questions. So, Andrew, when do you think you'll have some data on your hypothesis? Is it imminent or?
This year.
Yeah.
Yeah.
Excellent.
So. Yeah.
All right. Well,
not that hard with the experiments, but we're working on it. It depends on a few things, but within a year. Yeah.
and that's what
It's just a hypothesis. Just so
Yeah.
everyone knows. It's a hypothesis, but it's based on some pretty solid data that's leading up to it. So I think it's going to be right.
I think it's actually a really important piece of work. It's going to change a lot of what we think about COVID brain injury. Some of it is, direct, neuronal death. I mean, Danny's shown that, but it's not. There is a vascular component. There's a hypoperfusion element. There's an immune element. There's an autoimmune element. There's a mast cell activation element. There's a contribution from the gut biome element. It is several different pathways. But the one endpoint, and that's why I think Andy's work is so important, is what actually happens inside the cell. What actually happens to stop the cell from talking to the other cells, because that's what patients are complaining about.
Yeah.
Their cells aren't talking to each other, doc.
Yeah.
And so if we can find that, the virus itself interferes with transmission, that's a hugely important finding, because it's also a potential target for therapy. And I think that's the main thing.
all right. Well, we're out of time. Oh, yeah. One last question.
Sorry, you mentioned that you're starting this International Society for Long COVID, and I'm just wondering, how do you recommend we get updates about it? There.
I shall hand over to the incoming chapters.
Yeah.
President.
I don't know. I've been dragged into that, but
Yeah,
okay.
yeah.
But we're working on it very hard. We're going to present it at the conference in Amsterdam. We have a website up. We haven't advertised it yet, but there is a website up. I want a little bit more tweaking on it before I advertise it. But we've got we're going to have our founding meeting of the society on August 13th. So I got to get back from Australia first. So, okay. But thank you. We're moving along very nicely. So, and the more people that volunteer and help, the better.
Yeah, that's really great about the International Society for Long COVID. I'd like to just thank our speakers, Andrew and David. Thank you so much. And thanks to all of you for listening to the talk, and see you at the next seminar. Thank you.
Thank you.
Thank you for having us.
Pleasure.
Always.